Happy Saturday, scientists. Let's review. As you can tell by the DNA bow tie, this week we're going to cover some biology. B1, or cell-level systems, is our topic, and this video is intended as an overview and a revision of the important parts of this topic. If you have questions, please leave a comment below and we'll do our best to respond. Let's get started. We'll begin by discussing the two large categories of cells, and then from there we're going to move into the processes that are happening in those cells and how those function. So as we continue, you'll note that cells are divided into prokaryotic and eukaryotic cells. Those are the two major categories of cells. The prokaryotic were the first to evolve, and that's because they're a little bit simpler. They have no nucleus in the center, whereas our eukaryotic cells do have a nucleus, and we'll discuss the details of the nucleus again if you don't remember what those are. Prokaryotic cells are generally smaller, and these are things like bacteria, for example. Our eukaryotic cells are usually larger, and that's plants and animals that you're going to find in this category here. Now that we know about those two types of cells, let's talk specifically about the organelles, or the subcellular structures that are critical to the function of the cell. You may have heard in some of the definitions of eukaryotic and prokaryotic that they talk about membrane-bound organelles, and they're specifically talking there about the nucleus and the mitochondria. So our nucleus is the site of the cell's activity control. It's where the DNA is contained. Your cell membrane is going to limit the cell, and it's going to control the substances that can come in and out. You might have heard the words semi-permeable or selectively permeable to describe the cell membrane. The cytoplasm is where you have all of your chemical reactions, and then your mitochondrion, or if you're talking about all of them, the mitochondria, are your site of cellular respiration, which is the energy production for the cell. And you should hopefully remember that cells use ATP. So it's actually ATP that we're talking about when we're talking about energy production in the cell. And then of course we have to talk about those organelles that are only in plants. Hopefully these are familiar to you. So for example, the chloroplast, which is where photosynthesis happens. That is the plant's way of producing food for themselves. The next structure is the permanent vacuole. You may not always hear the word permanent used to describe these. They are a site of storage for nutrients and water for the cell. But they're also able to help to support the structure of the cell by having that water pressure. Those plant cells will also be surrounded by a cell wall. And this cell wall is there for protection a little bit, but it is mainly there for structure and support. And you need to know that it is made of a chemical called cellulose. And it's interesting that humans can't digest cellulose. We'll move back into prokaryotic cells, and we'll talk specifically about bacteria. It's important that you remember that they are the smallest living things. And remember that when we say living in biology, we mean that they can do all the things in Mrs. Gren. So they can move, they can respire, they can sense their environments, they can grow, they can reproduce, they can excrete, and they need nutrition. So if something meets those 7 things, then it can be counted as living. There are a few bacteria that you should be aware of as examples of bacteria and that you might be asked about. The first of these is E. coli, which is commonly known for its role in most food poisonings, but it is also very useful in terms of genetic engineering. The next is a large category of bacteria. It's your Streptococcus bacteria. These are the ones that are involved in sore throats, but they are also involved in other infections as well. On the more useful side of things, we have the Streptomyces bacteria. These produce the antibiotic streptomycin, which is a very useful antibiotic that can be used to treat a number of different diseases. For those of you that are interested in some extra reading, there's a fascinating book on the discovery of that antibiotic called Experiment 11 that I would highly recommend to you. Bacteria have some specific cell features that are different to the other cells. The first of these important features is a cell wall. In this case, it is more for protection than support, so it's a little bit different than the plant cell wall, and it's also made of a different material called peptidoglycan. And it's used for protection of the bacterial cell. They also will protect themselves with a slime capsule surrounding that cell wall. So this is for protection, but it is also for the ability to stick to surfaces. And finally, bacteria have plasmids, and these are small sections of DNA they can be exchanged between bacteria. Now, this is a good thing for us because if we want to do genetic engineering, it means we can move those plasmids around. Let's take a quick look at how we would recognize these structures if we were looking at these on a microscope image. So there on the left is your animal cell, and on the right is your plant cell. And we've labeled the nucleus, the cytoplasm, the mitochondria, and the cell membrane because those are the ones that are fairly easy to see. The cell membrane is a little bit more challenging in plant cells because it can be hidden right on the edge of the cell wall. So let's go ahead and label the parts that are plant cell only, as these are the ones that we often have a little bit more trouble with. So we'll have that cell wall, we'll have the chloroplasts, and depending on the details of the microscope image, you might actually see the inner structure to those chloroplasts. And then, of course, there is that vacuole as well in the center. But we need to talk about how we would get this type of image, and we would do that, generally speaking, using a light microscope. So let's go through and refresh ourselves with the different parts that are in a light microscope. At the top of the light microscope, you have your eyepiece. Underneath that is your body tube. And then you have the revolving nose piece, which I always find sounds rather amusing. And the revolving nose piece is carrying the various different lenses that you might have attached to your microscope. That's going to be suspended over the stage, and most stages will also have some stage clips to hold the slide in place. There will be some form of light source, And then there are your two focus adjustments. The smaller one is known as the fine focus, and the larger one is the coarse focus. And you'll use these two to move the stage up and down and to focus on what you are trying to do. So let's talk about how we would go about observing those cells. Remember that not only do you need to know the steps, but you also need to know why we do the steps. We always begin by lowering the stage down and by selecting the lowest power lens. And we do this to make sure that there is enough space and that we're not going to bump into anything as we go. Then you place your slide into where you're going to observe it. And then you raise the stage up to the top. Being on the lowest power lens, you have the space to do this, and this is the easiest way of focusing. So once your stage is up at the top, you're going to move into focusing. So you're going to look through the microscope, and you're going to begin by using the coarse focus. To lower the stage into focus. And you may need to use the fine focus for some extra adjustments and just to make sure. Once, of course, you've got this all in focus, then you might want to see those cells in greater detail, and so you might switch to a higher power lens. But when you do that, you want to make sure that you don't use the coarse focus. You want to only use the fine focus so that you are not going to worry about having your lens go through the stage and ruining both. So what exactly are you going to be able to see now that you've got your steps for using your microscope? Well, it turns out not a lot actually, because most cells and most cell parts don't really show up very well on a microscope. So in order to see them properly, we use our stains. There are three main stains that you need to know, and they each work for different things. Iodine is the stain that you use for plant cells because it reacts with starch. Crystal violet will react with bacterial cell walls. And methylene blue will react with animal cell nuclei. And so when you use these stains, you're able to get a much better, much clearer image. Now, from there we go into one of the few calculations that we need to do in this section of biology, and that is your calculation of magnification. There are two subsections to this. One is calculating the magnification of the image, and the other is calculating the magnification that the microscope is giving you. And it's easier to start with the microscope one. In this case, the magnification is equal to the power of the objective lens multiplied by the power of the eyepiece. And so any given microscope will probably only have one eyepiece lens, but multiple objective lenses, and so it might have multiple magnifications available. The other type is when you are dealing with images that have been taken on a microscope or drawn in a book. And so this magnification is equal to the image size divided by the real size of the cell. But remember that the units here have to match. And so often what you will be given is an image size in millimeters and a cell size in micrometers. So you need to remember that millimeters are 10 to the minus 3 meters, and micrometers are 10 to the minus 6 meters. So if you do that conversion, then you are able to have both your units matching and you're able to get the correct answer. But light microscopes aren't the only type of microscope that we have access to. We also have electron microscopes available. These are a little bit different than light microscopes in that they use electrons rather than light to visualize the sample. There's a tube-like structure generally, and then at the base there's either a viewport or some form of detector. At the top there's an electron source. This electron source produces a beam of electrons that go down through the tube, and they're held in focus by electromagnetic lenses. The beam of electrons will then pass through the specimen that you're observing. And the image will be detected. In many cases, this detector is connected to a computer to do some processing on the image, and often there will be some form of program that creates a false color image. These images are where the computer program adds color to certain parts of the cell to make them clearer to see. There are two different types of electron microscope. The first one is the transmitting electron microscope. And the second one is the scanning electron microscope. These are often referred to by their acronyms, so TEM and SEM. In the transmitting electron microscope, you get a much higher resolution even than the SEM, and certainly much higher than light microscope. The TEM will produce a 2D image that you can use. In the scanning electron microscope, you get a 3D image of the cell. For both of these types, you are not able to observe living cells, so the sample that you are using has to be dead. One of the things that we were able to observe when we finally got these microscopes set up is that we were able to observe DNA, and we had enough resolution to see that. And I suppose I should talk about what resolution is. Resolution is the smallest distance between 2 points where you can still distinguish those 2 points. You can still see that they are in fact 2 separate points. And I know that can seem like a little bit of a confusing definition, so let's try and look at an example here. If we have 2 points and we separate them, say, by about 1 centimeter, and if that was the smallest distance between those 2 points where you could still tell that they were in fact 2 separate points, then that would be a resolution of 1 centimeter. In the electron microscopes, we're able to get a very, very high resolution down into the micrometer range and even further than that, depending on the power of the microscope. And so we could see structures, for example DNA, and start to understand how it actually works within the cell. DNA is, as we know, the sort of code of life, and it is a polymer molecule. And it is made up of a monomer called a nucleotide. The nucleotide is made up of a phosphate group, which is connected to a sugar, and the sugar in this case is deoxyribose, which gives DNA the D in its name, and then that sugar is connected to a base. And that sugar will also be connected onto the next phosphate group, and so on and so forth, and that forms what we call the sugar-phosphate backbone of a DNA molecule. But it's the bases that are much more interesting for us. There are 4 options, A, T, C, and G, and the A and T are linked together, and C and G are always linked together. It is the order of the bases that gives us the sequence of DNA and that allows DNA to be the code for proteins. So the sequence would be the smallest level, and then the sequence of bases will be grouped together in genes, which code for a protein, and then a group of genes and other non-coding regions will be grouped together on a chromosome, and that's what you have in your cells. So what exactly do these genes and other things code for? Well, they code for proteins mainly. Proteins are long chains of a chemical called amino acid. And there are different types of amino acids, and by joining them together in a different order, you get a different protein. Many of the proteins in our body are actually enzymes. And so we need to link in a little bit of chemistry knowledge here. We need to remember that when enzymes are described as biological catalysts, then that term catalyst actually comes from chemistry. With our chemistry knowledge, we hopefully remember that a catalyst is something that is able to lower the activation energy. So if we have our reaction graph and we have a certain activation energy, using a catalyst in that reaction will lower the activation energy needed for that reaction. The next thing you need to know about enzymes is the theory of enzyme specificity, and this is sometimes called the lock and key hypothesis. What this means is that each enzyme is specific to the substrate that it is either breaking down or assembling. With the lock and key hypothesis, if we have, let's say, this as the shape of our substrate that the enzyme's working on, then the active site for that enzyme would have the exact same shape, so this kind of characteristic triangle in this case. And the chemical bonds holding that together are very important, and that's what will have an effect when other things come into play. So one of the main ones is temperature. So if we look at rate of reaction as we increase the temperature, and we need to bring back our chemistry knowledge again. So we know that as molecules move faster, the rate of a chemical reaction increases, and in enzymes it will reach a maximum and then it will rapidly drop off. This is because what's happened is that the enzyme has become denatured. The active site has changed shape because some of the chemical bonds have been broken. We have a similar pattern if we're dealing with the rate of reaction as a function of the pH. An enzyme will have an optimum pH level, and that optimum pH level is going to be different for different enzymes, and there are some enzymes that are designed for higher pHs and some that are designed for lower. For example, in the stomach with a pH of around 1 or 2, then the enzyme's going to have its optimum pH much lower than an enzyme that is not designed for use in the stomach. The next thing we need to know is we need to look at how the concentration of the enzymes would affect the rate of reaction. And again here we can put in some logic. If you think that as you increase the number of enzymes available, then they can increase their rate of reaction because they're able to process more substrates. But at a certain point that rate will drop off, and that's because they have used up all the substrate. The substrate concentration we can again try and see logically. As you increase the number of substrates available, then you increase the rate of reaction. But once every single enzyme is working and all of the active sites are full, then that rate levels off, and this is known as the saturation point. So every active site is filled with substrate, and so even if you increase the substrate, you can't actually increase the rate of reaction anymore. So what do we use these enzymes for? Well, one of the things we use them for in our bodies is to break down the food molecules that we have. So we know about carbohydrates, an example of which is starch, and then proteins are another large category of foods, and these are made up of amino acids, and lipid molecules, and these are made of 3 fatty acid molecules, and then your glycerol at the top. So if we take our carbohydrate, and we break it down into a simple sugar, we're going to need an enzyme called carbohydrase. To break down your starch into glucose, you're going to use an enzyme named amylase. And to break down your protein into amino acids, you're going to need an enzyme called protease. Hopefully you're starting to see that there's a little bit of a pattern here with this ase ending. These ase endings generally indicate enzymes. And if we had to take a guess for the lipids, well, hopefully you'd guess lipase. Now that we know about enzymes and the conditions that affect them, we can begin to understand some of our body processes. And one of the most important of these processes is the process of respiration, or how our cells get their energy. Respiration can be divided into two categories: aerobic respiration and anaerobic respiration. Aerobic respiration involves oxygen. In fact, oxygen is required for this type of respiration to work, whereas anaerobic respiration does not. In both cases, we're going to begin with glucose, C6H12O6, and in aerobic respiration, we'll react it with 6 molecules of oxygen to produce 6 molecules of carbon dioxide and 6 molecules of water. In anaerobic respiration, oxygen is not present, and so we'll still start with our glucose, and in our cells we'll turn it into lactic acid. Lactic acid is unfortunately toxic, and so this is not a product that we can have in our muscles long term, and so that's why you can end up with cramps when you're exercising, because you've gone into anaerobic respiration. There are some organisms that can do a process called fermentation, and this is a process that is also anaerobic, and the organism will take glucose and turn it into ethanol and carbon dioxide. This can be useful to us as humans, as both ethanol and carbon dioxide can occasionally be considered useful products. For example, if you were trying to make beer, you would use yeast to get the alcohol, the ethanol, and the carbon dioxide would give you the bubbles in the beer. You would also be able to use this process in order to raise bread, for example, where the carbon dioxide will help to leaven the dough. The reverse of the respiration reaction is known as photosynthesis, and this is the reaction that happens in plants. The photosynthesis reaction is exactly the opposite. So carbon dioxide will be reacted with water, and this will produce glucose and oxygen. The plants consider oxygen to be a waste product, but it's quite useful to us. The glucose will be used, for example, in starches, and that's a long-term store of energy for the plant. Cellulose for the plant's structure. The photosynthesis reaction happens in two stages: the light-dependent stage and the light-independent stage. In the light-dependent stage, it's an endothermic reaction. The energy from light is required, and it will be used to split up the water molecule. After that, the light-independent stage will be a reaction between the hydrogen that you've just produced from your splitting up of the water molecule, and you will react it with carbon dioxide in order to form the glucose. Because this reaction is so critical and has produced most of our energy as well as most of our oxygen, it has been extensively studied, and you'll need to know these experiments. Many of them involve the process of testing a leaf for starch, and to do that you will need to kill the leaf using boiling water and then remove all the chlorophyll using hot ethanol. You'll then wash the leaf, and then you'll add some drops of iodine in order to react it with the starch. And so hopefully we remember that if you do that, the iodine, if it's a positive result, will give you that blue-black complex that it forms with the starch, and so that will let you know that starch is present and that photosynthesis has happened in that leaf. If it's negative, then the leaf won't change color. So one of the first experiments that they did with this was to prove that chlorophyll was in fact necessary for photosynthesis. And to do this experiment, you need a special type of leaf like the one that's shown on screen here, which is called a variegated leaf. And in these leaves, you'll see that there's a white section where actually chlorophyll is not present, and then a green section where chlorophyll is present. And so you would start by taking the plant that has these leaves attached and destarching it, so you leave it in the dark for a few days for it to use up any starch stores that it already had. And then you would leave it in the sunlight for a few hours to do some photosynthesis, and then you'll take the leaf and test it for starch. And you'll find that the section of the leaf where the chlorophyll is present, so that's where the leaf is green, is going to change color, and it's going to form that blue-black starchy complex, but the rest of the leaf will not. And you do a very similar process if you were trying to prove that light is necessary for photosynthesis. So you'll take an extremely well-drawn leaf. There's a reason I am not an art teacher. And you would place a piece of cardstock over that leaf to block the sunlight from a section of the leaf. And then when you did the starch test on this leaf, you'd find that the areas that were covered by the card did not change color because there's no starch present in that section. To test for carbon dioxide, you're going to need to absorb the carbon dioxide, and we tend to use soda lime to do this. So you'll have your little plant here, again, very well drawn, and then you'd put some sort of container over it, and you'd have soda lime in there to absorb the carbon dioxide, and afterwards you'd see that actually there is no photosynthesis, there's no starch present because there's no carbon dioxide. Oxygen production in photosynthesis, it used to be tested in a very similar method that was actually quite cruel to the animals that were used. So you'd have a sealed container with an animal in it, some form of rat or other living creature, and a plant, and then another one with just the living creature. And the living creature in the second case would rapidly expire due to the inability to breathe because there is no oxygen in that case. The plant's not producing any for it. Nowadays we do this by using the chemical test for oxygen, which is to relight a glowing splint. And so you need to find typically an aquatic plant that will produce some bubbles. You capture those bubbles and then you test those bubbles with a glowing splint, and if it relights the splint, then that tells you that that reaction produced oxygen. There are a number of different factors that affect the rates of reactions in photosynthesis, and the first of those is light. And it reacts a little bit like a substrate does. So as you increase the light levels, the rate of photosynthesis will increase up until a point where it becomes limited by the other factors that affect photosynthesis. So it will plateau at a certain point. In terms of carbon dioxide, this is a substrate for photosynthesis, and so the graph that's produced in this case is the characteristic substrate graph. So as you increase the concentration of carbon dioxide, the reaction will increase. There we go, that's a better line. And that will reach a saturation point as shown by the X on the line. Photosynthesis is a temperature-dependent reaction, and so the rate of reaction will change with temperature, and there will be an optimum temperature for that enzyme, and there will be a point at which the enzyme will denature. Depending on the climate that a particular plant is from, it will have a different optimum temperature. And this is one of the reasons that scientists grow concerned over global climate change, is that if climates change and plants are not able to adapt to the new optimum temperatures, they may not be able to react properly. One of the experiments that might be used to do some studies on this is experiments on pondweed. In the classic example, you take a meter rule and you separate out a light source here with some little light rays coming out of it. There we go. And that light source will be separated from our beaker containing some pondweed and containing an upturned funnel so that we can capture the gas that is given off by our very lovely pondweed in there. So there's the little bubbles of gas coming out. And you'll find that as you decrease the light intensity, the rate of photosynthesis will decrease. So an example, if it took 2 minutes to make 10 bubbles, then our rate would be 1 divided by the 2.0 minutes, or 0.5 as the rate. So you need to be able to calculate these rates on the exam. Those of you who are taking the higher syllabus, you'll also need to be able to calculate light intensity using the inverse square law. What this law states is that the light intensity will go down with the square of the distance from the light source. So the formula is light intensity is 1 divided by distance squared. Another important topic on the higher syllabus is to be able to understand the interaction of the limiting factors for photosynthesis, and ultimately perhaps be able to apply them. And those of you who might want to go on into industry after this, well, this would be something that you'd apply to food production. So let's take a look at the rate of photosynthesis as it would be affected by light intensity. So we might do the reaction a couple times and we'll see that it levels off at a certain point, and maybe we did this at 2 different temperatures, at 20 and maybe 25 degrees Celsius. And we'll see that both of them actually follow the same line. And this indicates to us that the reaction is being limited by something else, because it's getting enough light and the rate isn't changing. So in this case, it's probably being limited by carbon dioxide because the temperature didn't have any effect. So the next thing we might do is we might increase the concentration of carbon dioxide. And so we do the reaction again, this time with a higher CO2 concentration around the plant, and our rate of reaction would go up. And if we were to repeat the reaction at both temperatures, we'd get 2 different lines. And so that would indicate that with that particular concentration of carbon dioxide, the limiting factor becomes temperature. So depending on the conditions, there are different limiting factors, and the interplay between these limiting factors is something that is managed extensively, for example, in industrial greenhouses, where we need to make sure that we can get the maximum food production while using the least resources. This is the end of our B1 topic. I know it was quite a long video, and that's because this is a very long topic. I hope that you found this useful and that you were able to continue your studies on this. There is a worksheet down below in the description if you'd like to take a look at that and try some practice questions. And next week I'll post the videos with the answers to that, as well as some discussion points on it. If you have any questions or comments, please don't hesitate to leave those below. In the meantime, happy sciencing.